Hi students, welcome back to online lectures. Today we are going to discuss about inhibitors of DNA replication and transcription. We studied about DNA replication and enzymology involved in replication and how DNA molecule replicates to produce daughter DNA molecules. Now in this session we have to study what are the inhibitors. Inhibitors are nothing but which inhibits the process. So inhibitors of DNA replication. So mostly to treat different kinds of the infections, different kinds of the drugs are administered into the bodies, into the human bodies. So where those drugs targets certain regions of the infectants. There are maybe bacteria or virus or protozoa and other kind of the microorganisms. So here the drugs which are used to control the infection are of different types. Some drugs may inhibit replication, some drugs may inhibit transcription, some drugs may inhibit protein synthesis in such a way. So in our syllabus we have to study inhibitors of DNA replication. So bacterial DNA guidase, that is type 2 topoisomerase, which is involved in the unwinding of supercoils during the DNA replication. Then only it is possible to separate the strands by helicase and followed by replication. So here the DNA replication is inhibited by mitomycin and nalidixic acid. These two drugs involved in inhibition of the bacterial DNA guidase. Likewise, another inhibitor, ciprofloxacin, which interferes with the DNA breakage and rejoining process. Likewise, mammalian topoisomerases are inhibited by etoposide and actinomycin drugs, which are used as anti-cancerous drugs. Likewise, nucleoside analogs also inhibit replication and are used as anti-cancerous drugs. So these are the different types of the drugs used in the regulation and inhibition of the replication, thereby used in the treatment of certain kind of the infections. Coming to next, inhibitors of transcription. So transcription is what is transcription? Synthesis of mRNA. From that mRNA protein is translated. Now those proteins are used by the bacteria or the viruses to carry out their life cycle. So to treat such kind of the infection, some drugs are used to inhibit such process are rifampicin, which binds with beta subunit of prokaryotic RNA polymerase and inhibits that enzyme. It is an inhibitor of prokaryotic transcription initiation. It binds only to bacterial RNA polymerase but not to eukaryotic RNA polymerase. That means it only acts on the bacterial RNA polymerase but not the eukaryotic RNA polymerase. Thereby it is safe to give human bodies. Therefore, rifampicin is powerful drug for treatment of bacterial infections, used for the treatment of tuberculosis and leprosy. Another drug, mitomycin, which intercalates with the DNA strand. That means it interacts with the DNA strand and it blocks the transcription. And this drug used in the cancer treatment. So thereby it is known as anti-cancerous drug. Another drug which inhibits transcription are alpha-amanitin. This alpha-amanitin is a molecule made from the death cap mushroom. You can see the picture also here. This mushroom is known as death cap mushroom and is known as potent inhibitor of RNA polymerase. So alpha-amanitin is extracted from this death cap mushroom. One single mushroom could very easily lead to fast death in 10 days. So highly poisonous mushroom from which this drug is isolated, which inhibits the RNA polymerase. The mechanism of action is that alpha-amanitin inhibits RNA polymerase 2 at both the initiation and elongation states of transcription. Coming to mechanism of action of actinomycin D. Actinomycin D intercalates with DNA strands. Actinomycin inhibits both DNA synthesis and RNA synthesis by blocking chain elongation. They interact with GC base pairs as they require the 2 amino group of guanine for binding. So actinomycin binds to GC base pairs. Why? Because it requires amino group. So which is found on the second position of the guanine nucleotide. So it interrelates the DNA and inhibits the DNA transcription. Actinomycins are used as anti-cancerous drugs. So these are all of some of the drugs which are used to inhibit the replication and transcription of different kinds of the infections. Thank you.
Bacterial proteins are synthesized in bacterial ribosomes, which are composed of two compartments, 30S and 50S subunits, that interact together to form a 70S complex. In the 70S complex, the bacterial messenger RNA and tRNA coordinate together to produce an elongated polypeptide chain. Different antibiotics work through the inhibition of bacterial protein synthesis. For example, tetracycline inhibits the binding of amino acid tRNA to the 30S subunit, therefore inhibiting the first step in polypeptide chain formation. Aminoglycosides bind to the 30S subunit and distort it. This distortion of the 30S subunit inhibits the binding of 50S to the 30S subunit and halts the formation of a 70S ribosomal complex. Chloramphenicol inhibits the peptidyl transferase enzymes, and as a result, chloramphenicol interferes with polypeptide chain elongation. Macrolides and clindamycin bind to the 50S subunit and inhibit the translocation of amino acid tRNA on the bacterial ribosome, thus inhibiting the elongation of the polypeptide chain.